Hey guys, we are into the 18th session and today's discussion will be about the overview of data analysis. The key objects of data analysis includes we have to first plan the analysis, program the crude analysis, then deal with chance, bias and third factors. We have to assess the casualties and we have to measure clinical or public health impact. The sequence of data analysis includes first we have to identify the study type, identify main variables, become familiar with the data, characterize the study population, examine the outcome or exposure associations, create additional two-way tables and conduct the advanced analysis. First, we have to study the data type and this includes this would be done in the analysis part itself, that is whether it is descriptive study or it is an analytical study, and we have to get familiar with the study, what the protocol is, what the questionnaire is, what was the analysis plan which was planned, what is the data collection procedures, and we have to obtain the electronic databases and decide on the software for analysis. This is how the analysis plan. From the nature of the research, if we are describing one group and if it is an acute group, we would have done a cohort or surveillance study and we may be measuring the incidence. If it is a chronic and describing one group, it would be cross-sectional survey and we would be finding out the prevalence. If it is analytical, that if we are comparing two groups, and if it is an acute and frequent, then we would have done the cohort study. We would be requiring for the relative risk and risk ratio. And if it is a rare and acute disease, we would have done a case control study and we will be having to find out in the odds ratio. And if it is in chronic and frequent study, we would have used cross-sectional study and we will find the prevalence ratio. If it is a rare chronic disease, and we would be doing the prevalent case control study and we will find out the prevalence odds ratio. And when you hear for the first time, it is difficult to catch it up, and we would have provided the PPT, the link of the PPT in the description, where you can go and download this and see then and then to get what it exactly means. This is actually a gist of all the sessions which we have seen till now. And then we have to identify the main variables. What is the outcome variable, what is the exposure, what is the potential third factor, and what are the variables of the subgroups. Then we have to become familiar with the data. We have to produce the frequency distribution. We have to conduct the descriptive statistics, like all the variables describing the study population has to be studied. Then we have to review the number of observation by status and database, like we have to look for duplicates or missed observations. Then we have to check the range and legal values and check the consistency. Then we have to characterize the study population, like baseline characteristics, then age, gender, sex, like that. Then we have to find out the frequency of the clinical features or the health problems, and then in the analytical study for the compared groups, we have to do for both of them. Then we have to examine the outcome or the exposure associations based on the hypothesis called as priori. That is, we have to compare the groups for frequency of exposures using appropriate measure of association. Then we have to examine the association with the prior knowledge and the study design. We have to then create additional two-way tables which we will be requiring for our analysis, and then we have to go for the advanced analysis, that is dose response, stratifications, multivariate analysis, that we will be doing based upon the study to study basis. And some practical tips for the data analysis includes we have to first prepare the data analysis in advance. Then we have to analyze by stages like recoding, descriptive, analytical. All the stages we have to analyze, and we have to avoid post hoc analysis and data dredging. The initial stages of analysis includes recoding stage and descriptive stage. Recoding stage is creating the outcome data and recoding the key variables. Descriptive stage is calculating the frequency of the outcome. The analytical stage consists of univariate analysis, like frequency of outcome by age, gender, income, or outcome by income categories, like that. Stratified analysis includes frequency of outcome by income, stratified for age, gender, and several other things. Multivariate analysis includes logistic regression model. There are some of the softwares for data management like Epilinfo, and these softwares we can use, and there is an advantage that we can avoid the spreadsheets for data management or analysis of any type size. So this is a helpful software. Thank you. Our next discussion will be about the ethical framework for health research.